Good morning. Thank you for having me here today. My talk will be a version of the one I gave last month at AGBT, titled The Unified Whole Genome and Exome Automated Workflow, Suitable for High-Throughput Processing and Quality Assurance of Frozen and Formalin-Fixed Paraffin-Embedded FFPE Clinical Cancer Samples. Carolina Bocanegra was also a co-author who went with me to the conference, but she was unable to be here today, so it's just me giving the presentation. Before I get into the actual workflow, I'll just give a brief overview of the Genome Research Platform. So our role here at OICR is to provide next-generation sequencing and cancer genomics capabilities to researchers in Ontario, but also internationally. Although our focus at OICR is on cancer genomics, we have worked with other researchers in other areas such as viral evolution and primate genomics. We offer DNA and RNA sample preparation, sometimes also custom library construction, as well as sequencing services. We do have a lot of experience in a variety of sample types, including tumors and metronormal tissues, xenografts, cell lines, and as well as circulating nucleic acids. We do evaluate and adopt new technologies depending on what the collaborator is looking for. And if no new technologies or methods exist, we do work on any customized options. We do have a variety of options at GRP. We can focus on small pilot studies, but our focus of this talk will be genomics at scale. So now with the rapid advancements in technologies, we are now able to really sequence a larger number of whole genomes and whole exomes. So now the problem is starting to become, the bottleneck is starting to become rather library preparation. So at GRP, we actually use a robotics and automated workflows, such as this image here, which is a Cyclone NGS instrument. And this really lets us process large-scale multi-year studies using automation and robotics. And this allows us to keep the results consistent across users and across a large time frame. And it also allows you to reduce the processing time for your research assays and increase the productivity as well. But however, it's not just the volume that we're after. We do have to keep in mind that you have to ensure the libraries are sufficient quality, or else you keep on going back to do top-ups, which is really not efficient. And this brings us to the unified whole genome sequencing and whole exome sequencing automated workflow. Here we created an automated solution that integrates both of these into a single library prep workflow. And the cool thing that we added here is MiSeq shallow whole genome sequencing as part of the quality assurance system to ensure that the library, before it actually is enriched, is of high enough quality, since the probes are actually quite expensive. So I'll just briefly go over the workflow before I show some of the data. So at the start, we begin with our DNA, which we then quantify using a fragment analyzer to make sure it's of sufficient quality. We then proceed with mechanical shearing using the Covaris plate instrument, which will shear one sample at a time, but it's capable of handling plates of 96. And we have found that depending on the sample type, different conditions have to be adapted. FFPE will shear to a different size, even if you use the same program as a normal. After the shearing is complete, we do take an aliquot of it and then confirm the sizing based on a fragment analyzer. And then we proceed to creating a whole genome library using the Kaplan hyperprep. Once that is complete, we enter another fragment analyzer, and then we do shallow whole genome sequencing. If the collaborator just wanted a whole genome at this point, we could then skip right through production scale NovaSeq sequencing. If, however, the desire is to go into more targeted enrichment, we will then use the metrics of the shallow whole genome sequencing to ensure that the library is of sufficient quality before actually proceeding into pooling. So based on the metrics we get, such as percent match, percent coverage, insert size, we can then pool the samples. For this project, we did 4-plex, but we can do 2-plex, 8-plex. Just depending on what the collaborator desires. We will use, in this work, we will use the XGen hybrid capture. Once enrichment is complete, we then take another fragment analyzer and then do a low-pass sequencing as well. And this low-pass sequencing in the MiSeq stage is really used to determine how to really balance the production grade flow cell. So this is some of the data we have for this project. So we ran 266 libraries across different days on the Cyclone G3 NGS workstation. And we can see that it shows uniform insert size, percent mapped, and total reads across 6 different runs. And then I'll go over how the MiSeq shallow whole genome sequencing data is actually indicative of how it will perform. So this here is really what we've used to move to a MiSeq as a pre-NovaSeq QC. So we can see that there's a high predictability between coverage per gigabase on MiSeq versus a NovaSeq, which we can then use as a model to determine the optimum number of samples per NovaSeq run. And this is consistent across fresh frozen and as well as FFPE sample types. We've also seen a high correlation between the number of sequencing reads generated and the resulting coverage on NovaSeq 6000. So since the reads per sample is highly correlated per coverage, we will then use these metrics along with the shallow whole genome sequencing to really determine how much sample you can load before you actually move into production grade sequencing on the NovaSeq platform. So this entire workflow is actually automated using the robotic system, including even the washes and the hybrid capture, which is actually one of, I find, the most labor-intensive part. The kappa whole genome is relatively easy. You can do it in about a day. But this part actually does take a lot of work with a lot of different washes and beads cleanups. We can see the distribution of percent on target and insert mean for each captured pool is fairly consistent. And it also, from the MiSeq to the NovaSeq, we can see that it provides a very close alignment. And this actually has a better indication. So in the blue, we have the MiSeq on target rate, a 10x coverage. And then in the orange, we have the NovaSeq on target at 5000x coverage. And here, this lets us see that the MiSeq QC metrics can be used as predictors for how the NovaSeq will function. So if we adjust the MiSeq percentage on target with around a observed 10 to 15% loss, we can then determine that we'll need to increase the loading of the sample to compensate for this loss in percent on target. So some of the conclusions we've seen from this talk. So a high-throughput automated whole genome and whole exome workflow is capable of delivering high-quality metrics at scale. It delivers a uniform percent on target as well as coverage across runs. So what it really does provide is a lot of flexibility for researchers in the future. So if you just want a whole genome library, we'll create the whole genome and then go into the MiSeq, then to the NovaSeq. But if in the future you want to get a deeper coverage on a certain area, we can then go back to this exact same library and do a target enrichment with the same sample type. So this really reduces any variability that you might see instead of traditional methods where you'd have to go back and re-extract the sample and create an exome or targeted sequencing from another library. The MiSeq shallow whole genome sequencing is essential for a pre-NovaSeq predictor, just because the NovaSeq is actually very expensive to run, and this actually lets us predict how the NovaSeq will perform before we actually run the instrument. And then again, it helps us identify and exclude any poor quality libraries from the costly production scale sequencing. Then I'd like to acknowledge my fellow co-authors that helped put this poster for AGBT and as well as the whole genome research platform. Thank you.